3 by Martin Buber, Ich und Du, I and Thou. And Buber had the claim in this book that the world is given to us in two fundamentally different ways, that we are interacting with the physical world in an I-it relationship. These are things that we can manipulate and interact with in a way that's fundamentally different from the I-thou interaction that we can have with other human beings. And I was just reminded recently of this when I talked to my colleague Bruce McEwen, who's shown you on the left-hand side. This is not me. We had lunch together, and he was pointing me to an article that was published about Buber. But we often talk about Buber because he named one of his dogs Buber. So this is Buber. And so I like this picture because you can see the sort of interaction between them, this I-thou or I-you interaction between them. We also call this a second person interaction because thou would be a second person pronoun. And when we are watching this, it's a third person interaction. So today I'm going to talk about faces, what we know about face perception and the neural circuits supporting them, a little bit about context of faces like bodies, but then also about interactions between agents that are made of faces and of bodies. So primate sociality is of course built on general mammalian sociality, but primate sociality is special. And primates are social intelligent individuals. So I'd like to show this movie here that's symbolizing how our lives are revolving around each other. But it's also showing, even if you don't know these particular macaque monkeys, which are Tonkean macaques, you can kind of get the story, understand the little social drama unfolding here. And I'm always showing this then as a second sequence. So someone just invented a very important tool here, the nose pick. And the reason I'm showing this is that there are two theories about what made primates smart. The first theory is the social intelligence hypothesis. The idea is that primates had to become smarter and smarter because the social environments became more complex, and those who were better able to predict what was going to happen in these social complex environments would have an advantage over those who could not. The other theory is that because of the anatomy of the hand, primates are better able to manipulate the environment and use it then also to invent things as important as the nose pick. So as anyone's guess then, what really made us intelligent, invention of the nose pick or social understanding. Primate ethologists have argued for a long time that primates possess a very intricate knowledge of their social environment. So in baboon monkeys, for example, it's thought to be roughly in 3 different levels of organization. So the first level is knowledge of other individuals in a social group that can usually involve maybe like 100 individuals or so. And primates would know like the age or the gender of individuals in this group. Second, primates are very keen observers of the social interactions that are taking place between these individuals. So interactions could be grooming, mothering, fighting, different kinds of social interactions. And based on these 2 levels of understanding of the social environment, they're constructing high-level representations of their social environment like friendship, kinship, or hierarchy of their social environment. So this, to me, is very interesting because you have to represent these kinds of representations as a graph. So if you know that A is the mother of B, you know that B is not the mother of A. So this kind of knowledge is not just associative, but it has a structure to it. And so I would love to understand eventually like how such knowledge representations could exist in the brain, but we are still quite far away from it. So social perception and social cognition really start with the processing of individuals, and this is also where I will start, and that's namely with the face. So faces provide a huge amount of socially relevant information to us, like information about the mood, attention, species, race, gender, age, identity, familiarity, attractiveness, trustworthiness or perceived trustworthiness. And you get all this information from a brief glance at a face that can be a fraction of a second. So even faster than I can just even read through this list of information. And so you get this through a face processing system. But in order for this to work, the system has to solve several computational problems. So if you have a social scene like this one here, it's not going to be difficult for you to figure out that there are faces there, that there are 3 faces there, where the faces are, even though the lighting conditions are very bad, even though the faces can be close or far away, therefore have different retinal sizes, head orientations can be different, and so on and so forth. But detecting faces, knowing that there are faces, knowing where the faces are, is relatively straightforward for us. But then if you have seen Godfather 1, you will also know that these 2 pictures are pictures of the same individual, even though on a pixel-by-pixel basis, these 2 pictures are, of course, much more similar to each other. So what does it mean? It means that there's something happening in our brains automatically that is uncovering the cause behind these 2 images, that is a particular individual, even though the individual is now appearing at different orientations. And so we also like to take the face processing system as an example of sort of this causal inference. Just as a brief reminder, so there's about 1% of the general population suffering from a condition known as face blindness or prosopagnosia. To a face blind person, the social world might look something like this, where virtually all the faces look identical. Here they actually are identical. And so as you might gather, if you have this difficulty telling one person from another, you're running into all kinds of social problems in daily life. And many, therefore, who are suffering from face blindness, prosopagnosia, their enthusiasm for social interaction is really greatly curbed. So we have no good understanding right now, like, yeah, so there are two different categories. So there's developmental prosopagnosia, where we think right now they're likely born with this condition. There are not so many investigations of really like very early face recognition abilities and then long-term studies. But face recognition abilities are very strongly heritable, and so the belief right now is that there are genetic components even determining face recognition abilities. And then of course there's acquired, which we can understand. There are lesions to the brain. If they're affecting circuitry processing faces, then of course face recognition is going to be impaired. Okay, so what do we know about the basis of face recognition? It all started with Charles Gross in the late 1960s. He was recording from this part of the macaque monkey brain. Early vision starts here, but he knew from lesion studies that this part of the brain, known as the inferior temporal cortex, was critical for object recognition. And when he recorded from single cells in this region, he would find cells like this one here, which during the presentation of a face would increase the discharge rate, but not during the presentation of a control stimulus like a hand or other controls that he used, but not too many because it was difficult at the time to have these control stimuli. So he found evidence for single cells that are specialized in a particular category faces. And subsequently, several people like David Parrott, who was also a pioneer of this work, found face-selective cells pretty much distributed all over inferior temporal cortex. Every symbol here is marking a location where someone has found a face-selective unit. So the thought at the time was that face cells are intermingled with non-face-selective cells. You would find papers where more than 1,000 cells would be recorded, and then about 10 of them or so would be face-selective. So it's very difficult even to find face-selective cells. So Nancy Kamesher, with the advent of fMRI and pretty much one of the first really very solidly done fMRI studies, she found an area, and now we know that there are at least 3, that are in the human brain that is responding more strongly to faces than to non-face objects. Now, fMRI is a very indirect measure of neural activity, measures changes in blood flow or oxygenation, and these maps that you typically see are thresholded significance maps, right? So it might just be that there are a few more face cells in this region inside than outside this region. But it was taken as evidence that maybe in the brain there are modules that are processing face information. And if you take the modularity account very strictly, you would even propose that maybe all that these areas are doing is to process face information. So how actually are faces processed or sound? I then thought we had to combine these two different approaches. We have to use fMRI to get an overview over the functional organization of the whole brain, and then use single cell recordings to learn more about how the cells are processing information. We used the human fMRI scanner, and then contrasted activation during presentation of monkey or human faces with activation during presentation of non-face control stimuli, and found, as shown here in this computerized map where the brain is inflated to show the cell size here in green, and the gyri, in dark gray, and the gyri in light gray, that there are 6 regions in the temporal lobe of macaque monkeys that are selectively responding more strongly to faces than to non-face stimuli. They are interspersed with regions that are responding more strongly to non-face stimuli than to face stimuli. These are the blue regions. We have these 6 regions on both hemispheres, so there's a total of 12 of these areas. Now, when we record from this area, we find that virtually all the cells in this fMRI-identified region are face selective. So I'm showing this here in this population response matrix, where we plot the cell number from top to bottom, stimulus number from left to right, the 16 faces in this image set on the left-hand side, and you can see immediately that the vast majority of cells is responding more strongly to faces than to any of the controlled stimuli. There's a smaller group here that's selectively suppressed, intermittent group where it's not clear what they're responding to, but the vast majority of cells are selectively responding more strongly to faces than to non-face objects. If we average activity across these different conditions, we find that the response to the faces is the strongest, but we find some of the non-face stimuli to elicit intermediate responses. And then if we look at what these stimuli are that give these intermediate responses, we find that these are stimuli that have things in common, visual features in common with faces. So these are typically roundish objects with a bilateral symmetry, an internal structure, and so they can sort of fool the face processing system to treat these stimuli as faces. And so I really love this picture of these sliced peppers because you understand that they are not faces, but you cannot stop yourself from seeing them as faces. You also know they've just been sliced, so they have reason to be really mad at you and scream or shout. It's impossible for you to not see that, right? So there's something in our brains that's wired that in a way that we want to see faces in the outside world. So what have we learned about the face processing system? Once that we know that there are all these face cells in all of these face selective areas, and this is true for all the areas we've recorded from so far, we can ask, like, why are they face selective? How are they processing faces? One of the most fundamental discoveries we made is that these cells are selectively tuned to facial features. So here's an example cell that is responding most strongly to the eyes in an almost cyclopean arrangement when they are close to each other, and then the firing rate is systematically reduced when the eyes are straddling the outside of the face. The same cell is responding also to big irises, not small irises, and so on and so forth. So it's almost as if the cell would take a ruler and measure these features and relay them in a very simple, meaningful, one-to-one mapping of these features in its output firing rate. Just as an intuition, this picture of this broad ram-shaped tuning curve easily explains fundamental human psychophysics of face perception. The idea is that face perception works similar to color perception, that there is a space of faces spanned by their physical properties with a center that would correspond to perception of a white color. And then if you're adapting to red color, for example, your perception is shifted in the opposite direction of the color space, and analogously also in face space. If you have these very broad tuning curves that go from one extreme to the other, you would easily explain this property of face space. Srivatsan-Sadagopan and Robert Zarkow inactivated one of the 12 face areas while the monkey was tasked on a computer screen to localize a face. This is the most fundamental face processing task, the detection of faces in a cluttered environment. And when they inactivated one of the 12 areas, they found that the behavior was impaired as if the contrast of the face that they would have to find would be reduced like this. It didn't happen for bodies, which we use as controls, because for every face area there's a body satellite area nearby. And also not for shoes, which we use for other reasons I'm not going to go into, as controls. So selectively impairment of even the most fundamental face processing by an activation of just one out of these 12 face selective areas. Sebastian Möller was interested in determining what is the relationship of these different face areas. He used electrical stimulation inside the MRI scanner without visual presentation, and he would place a stimulation electrode either inside a face area or outside of a face area. When he would place the stimulation electrode inside a face area, what he found was a selective activation at the location of the other face areas. So this is indicating that there are strong connections between the face areas, forming a face processing network. Actually, like, one of the puzzles for us is to understand, like, why there are these strong connections between the face areas, and the connections to the outside world actually are a little bit more mysterious than the ones that are grouping together the different face areas into a face processing network. So the primate brain has a dedicated face processing network. And it turns out that the different nodes in this network have different functional properties, which I'm indicating here by different colors. So for example, Pablo Polosecki and Clark Fisher, two gradsons in the lab, found a seventh face area that is selectively activated by natural facial movement. And also in the other areas, as indicated here by the different colors, we have different functional specializations. So in this face area here, which we called ML, we find face cells like this one here that are very strongly tuned to head orientation. So a given face cell might only respond to left profile, not to the front view, and not to the right view. If you go like one level higher up, the cells are still very strongly tuned to head orientation, but now they exhibit a very peculiar property that is mirror symmetric tuning. So a cell that is tuned to the left profile of the face is also tuned to the right profile of the face, but it's not responding to the intermediate head orientation, the front view. Sometimes they're even suppressed by the front view of the face. And then if you go yet one level higher, you now find cells that might not even care very much for different head orientations, but they can be very selectively tuned to particular facial identities or combinations of features. So what does it mean? We now know that there is a transformation of face information within the network, that this transformation occurs in two major steps from representations that are qualitatively, not just quantitatively, but qualitatively different. And that is a transformation from a representation that early on is more veridical to what is currently in your eye, what is the orientation of the face, to something that is more veridical to the cause that gave rise to this stimulus, that is who is in the picture. So transformation from a picture-based to an identity-based representations in two major steps. So this is very interesting also for theoretical reasons. I'm not going to go into this in great detail. I'm just going to give you some indications that even though faces are very complicated stimuli, because of this functional organization, because the primate brain has evolved this specialized circuitry, we can now understand face recognition at mechanistic level, and we are aspiring to identify the computational principles that are implemented by the system to achieve these transformations. So I already mentioned before that one of the things that are difficult for us to understand is this mirror symmetry representation here. And the reason is that when we found the mirror symmetry at this level of processing, and we found it in every time we've recorded from this area, there was no computation model that predicted that it should exist. The reason for this is intuitively clear, is that most learning rules that people use to wire up hierarchical information processing networks are temporal contiguity rules. So you're wiring together selectivity that is activated either coincidentally in time or in close succession in time. But when you see a face move, like my face when I'm pacing up and down here, and you see my face from the left or from the right, you will always see my left face rotate, and then you see the left profile with the front view, and then the front view and the right profile. So the intuition is that for a temporal contiguity learning rule, you would actually expect that the tuning curve would get broader, so to group together the left profile with the front view, but instead you're getting the left profile and the right profile view, even though they will never occur in temporal contiguity to each other. So Joe Leiber was a grad student with Tommy Poggio, for us to understand this better, built a very simple model system that captured the main organizational features of the network as we understand it. So he was saying the network has a modular architecture, which means that it's going to be filtering information that's on faces and suppressing information that is not on faces. We are positing here that level 3 of processing, that the system has 3 levels of processing. The first is the face filter. The last one has a computational goal that is to identify the person in the picture. And when we're applying a Hebbian learning rule, but only when we apply a particular version of the Hebbian learning rule, which is Oya's rule, we are getting the mirror symmetry at the second level of processing. We don't get it for any other learning rule that we looked at. So the fact that we find a particular tuning property at one particular location inside the network, if this is correct, I'm not claiming it is correct, but it's the only theory that we have so far that predicts the mirror symmetry, would be a consequence of the specific learning rule that was used to wire the system. It's also a consequence of the bilateral symmetry of the face, so the geometry of the stimulus, and of the organization of the system into these 3 levels of processing. Finally, I wanted to mention recent work that Kaspar Schwerzig, a postdoc in the lab, published, where we found evidence for feedback processing in the system in the form of predictive signals. So many of you will know that predictive coding has been proposed to be an efficient way for a neural system to not waste energy on things it already knows, but to respond more strongly to things that are not predicted. And so Kaspar used the organization of the face processing system to find that actually, yes, there is a prediction error in this particular area of the brain, but the properties of this prediction error signal in this region do not reflect the local tuning properties, but the tuning properties of these high-level areas. So the prediction error depends on identity, not head orientation, as a little bit of a reflection of mirror symmetry. So we had this transformation in 2 steps, and I was just briefly indicating one way to think about this, which is the classical view, to think that an observed image is mapped onto an identity, and that this is the task of the system to do this. And this is the assumption of most deep learning networks that are around these days. I just wanted to briefly indicate that maybe there's a different perspective, and this is work that we've been doing together with Ilke Yildirim, joint postdoc in Josh Tenbaum's lab and mine, where we thought that maybe what's happening is not a mapping on identity directly, but on shape and texture attributes of a 3-dimensional face model. So in theory, this is a very different way of looking at this mapping. What it means is that we will get a mapping of an observed image onto a 3-dimensional face model. If you have this mapping, if basically the activity of the cell at the most high level of this processing does not only tell you who it is in the image, but it will give you all the parameters of the face, including the head orientation and including the lighting condition, it means that you can fully explain what you are currently seeing in an image in terms of a fitted 3-dimensional face processing model. And it turns out that this model is actually better to explain the physiology that we have, and it's also better to explain human psychophysics than simple feed-forward networks. in the sense that it makes mistakes when humans make mistakes, and that it's better than artificial systems when humans are better. So one way to visualize it is that when we see an image like this one of Audrey Hepburn here, what we really see is not just a two-dimensional image, but what we are inferring is sort of like a three-dimensional model of her face, which also means that we can make predictions about how she would look like if lighting conditions would change, or if her head would rotate like is shown here. The particular model that makes particular predictions for the different processing areas, and this is important to us for future experimental tests of this model to have predictions that are qualitative in nature, and this is something that we're working on right now. So what have we learned? So number one is in the primate brain there's specialized circuitry for the processing of face information, and that's very likely because faces are so important to us. What it also means is that we can understand now something very complicated. It's the processing of stimuli that are as complex as faces, because there's a particular neural architecture in the primate brain that has about as many neurons in the face processing system as there are in the entire mouse brain. So in some ways our task to understand how the system is working is even easier than to understand the mouse brain, because we think that there's only a very limited number of computational purposes. These are only cortical areas. We can image them and therefore figure out what they're doing. And we can do this from the level of face cells. We have some indications now how we can explain even quantitatively human face recognition through this organization of face areas into face processing networks. Now, when I showed this slide before, I was implying a model of perception where there is a stimulus out there that contains implicit information, and that the perceptual system then goes in and is trying to extract this information from the face. In my title of my talk, I talked about the dual face. The face has sort of like a dualistic nature. And what I mean by this is that faces actually are not just passive tableaus for observers to gather information from, but they're actually very active social agents that are doing things to the observer. So here's one example. I've been showing this video of a 3-day-old macaque monkey for many years now. I've seen it hundreds of times. And initially I was showing it for an effect that's going to appear in the video in a second. But what it does invariably is that it makes us happy. It gives us like a warm, fuzzy feeling. And there's no way that we can avoid that reaction from happening. And I can testify to this because I've seen it, again, hundreds of times. Every time I see it, it's this warm, fuzzy feeling. Granted, it's not just the face, it's also the body, it's the towel, it's everything together, but it's just adorable. And so you have this response. What does it mean? That there's a particular pattern from a vision perspective, a 4-dimensional pattern of pixels that somehow gets into your emotional brain automatically, and there's no way that you can stop this from happening. This is the reason why I was showing the video initially. After birth, for about 2 or 3 weeks, macaque monkeys are imitating facial movements that are perceiving. And those of you who have children know that also in humans this occurs for about like 3 months after birth. Then this behavior goes away. So there is something specific in this little monkey's brain, even though it has very limited experience with the outside world, that is selectively processing facial information and then responding to it with a motor response. This is happening automatically. Another effect I can demonstrate to you. So with this pair of glasses. So you notice that these are not my real eyes, right? So my real eyes are still here. These are just painted eyes on glasses. What's happening is you're looking at me right now. My eyes seem to be looking to the left. Your attention is drawn in the direction of my eyes. You look over there, you see nothing happened. Your attention is drawn back to the face because faces attract attention. I still seem to be looking to the left-hand side, which I can assure you I'm not. Your attention is again drawn to the left. You realize there's nothing happening there. Back and forth, back and forth, back and forth. So over time, I have to take off the glasses because it becomes annoying. So what's happening here is that faces are manipulating our attentional system. And again, like once you paid attention over to the direction where I seem to have looked at, you could control your attentional focus and could move it back to where you want it to be. But for a brief moment in time, faces manipulated your attentional system. And finally, memory. If I show you a picture of Charles Darwin, you will not be able not to know that it is Charles Darwin. Even if I had not mentioned his name, you will not be able to not remember things that you know about him. So faces are not just passive tableaus of social information, but they're very active manipulators of perceivers. And so when I was studying at Rockefeller, I was thinking, okay, if we have a dedicated face processing network, what is it for? We don't really know like what it's projecting to, but somehow faces have this power to elicit emotional, mnemonic, attentional effects. So maybe there are strong projections from the system into these other parts of the social brain. And so we are looking for neural correlates of these higher level functions, cognitive functions, mnemonic functions, motor functions, emotional functions that might connect to the face processing system. In turn, I was also thinking that if we don't know how the information from the face processing system is used, then we don't really know its computational goals, and therefore we will actually not be able to understand the system. And so the last years we've been trying to map out circuitry, and I want to briefly talk about 3 of them in overview that we think are potential correlates of these different functions. Okay, so I'll start with something that is known as intuitive psychology, and I will motivate this in a second. So I showed you this video of interactions happening between tongue-tailed macaque monkeys, which you understood and told a story about, and I told you that primate ethologists have long been positing that primates are very keen observers of social interactions. So we thought, okay, we should understand like how social interactions are processed in the brain. Maybe it's not possible, maybe it's too complicated, but maybe it is possible, so we should try it out. One reason for that is that in the human brain, even very abstract individual elements, when they are interacting in an appropriate way, give rise to the illusion of a social interaction taking place. So in these famous Heider Zimmer movies from 1944, it looks as if there is a mean big triangle that's harassing a small triangle, that there's this circle sort of looking and trying not to be detected, trying to watch what's going on, trying to hide. Again, you cannot unsee this. There's no face here, there's no body here. It's just the interaction that's taking place between these geometric shapes that seems to imply that a social interaction is taking place. When you show this to human observers in the MRI scanner, the regions that are being activated while they're watching these social interactions versus controls that don't imply social interactions are regions that are otherwise activated in the most high-level forms of social cognition when you're making mental state attributions to others. So it's medial prefrontal cortex, the temporal pole, and the temporal parietal junction in the seminal study. So we thought, so maybe if you're processing social interactions, there's a way to get into high-level social cognition circuitry. So we had different hypotheses about what would happen if we would look for social interaction processing. And again, this is third-person interaction processing. There's an observer seeing 2 others interact with each other. So one was that maybe regions in the face processing system that are more involved in face motion and neighboring areas involved in body motion, that they would be more engaged because, after all, an interaction is a specific kind of motion. We thought that maybe the anterior parts of the face processing system might be more engaged because we had found before that the context of bodies influences the processing of faces in these areas more than in posterior parts of the system. And social interactions you can think of as a broader context for the processing of face information. We thought about the mirror neuron system, a parietal premotor system, indicating the 2 main areas here. Why? Because it has been thought that this system, through the process of simulating actions of others who we are observing, gives us access to the intentions that are behind the actions that we can see, again, through the process of simulation, because we know our own intentions when we do the corresponding movement. If you think about an interaction, like a goal-directed behavior, it's an interaction between an agent and an object. And so maybe also social interactions between agent and agent might engage this particular system. And finally, maybe even in the monkey brain, we could find potential homologs or precursors of those areas that in the human brain are responsible for theory of mind or intuitive psychology, so the understanding of others in terms of their hidden mental states that we will never be able to see or feel or touch, but this is the way we interpret the actions. So what we did was use the same logic with fMRI to find areas of specialization, but now not for faces, but for the processing of social interactions. And when I say we, this was all the work of Julia Sleever, an amazing postdoc in the lab. So she showed videos, 3-second clips of videos from different categories, including social interactions. She had a split screen arrangement where there are also 2 monkeys, but each of these monkeys is engaged in a separate non-social goal-directed behavior like drinking or picking food. We had sort of still controls. These were movies of agents that were not engaged in obvious goal-directed behavior. They would move a little bit. And then we had physical controls, toys that the monkeys use, either bounce into each other, moving separately, as shown here, or still. And so from these videos, Julia could then construct particular comparisons between the different video contrasts, different video categories, to exert information about, okay, which brain areas are active when there are just agents present. So monkeys with faces and bodies. What happens when there's now activity when there's no activity in the observed animal, there's a goal-directed action beyond just the presence of the agents, what happens when there's a social interaction taking place, or what happens when there's a physical interaction taking place. These are some videos, some social videos of actual social interactions that he was showing. Here are scrambled versions that she used as a control. And Julia spent a lot of time marking in every video frame where's the face, where's the body, where's the gaze of the monkey going to, and then where's the subject monkey looking at when he's watching that video. The reason for that is that, of course, the analysis of actual real video in fMRI is very difficult. And so this allowed Julia to form nuisance regressors that we could then take out from the activations that we found. So whenever I'm talking about any activation in the following slides, we took out motion energy, luminance, face, body, hands, velocity, behavioral features like where the monkey is looking on the screen, and interactions of the two, like whether the subject monkey is now looking at a hand or a face or a body. Okay, so what did she find? So here, showing the face, the body, and the object areas in yellow, pink, and green, she looked at the activation by the presence of agents, which consists of faces and bodies. And not surprisingly, she found activation in face and body areas, both in the temporal lobe, but also in the frontal lobe, which I didn't mention before. We have 3 face-selective regions in the frontal lobe as well. When when she now looked at actions versus no actions, so the observation of goal-directed behavior in others, she found a selective activation of the body areas, but not of the face areas, even though the face and the body areas are always coming in pairs. So, and even though these areas are just defined by whether they respond selectively to faces and then or to bodies, when the monkey is observing a goal-directed behavior, the body areas are much more activated than when they just see bodies or an agent in isolation. When she looked for social interactions versus actions, now the face areas are also much more strongly activated, and the body areas as well. So there's a functional dissociation between face and body areas, even though they're nearby, that we cannot explain at this point by low-level visual properties, but by more high-level properties, i.e. the presence of faces in a social interaction video. What about the mirror system? Julia mapped it separately, just as she mapped these areas separately. She found that yes, there is engagement of the mirror neuron system by social interactions more than by no-action controls. She also found, and this is maybe the most interesting finding on the mirror neuron system, that the system is completely engaged by just observing physical interactions between physical items. This is important because there's no agent there, right? So there's no intention that you will infer when you're watching this. So we think that maybe the mirror neuron system is engaged more broadly in the inference of causality or the processing of interactions, both physical and social. And when she looked for social interaction versus all the control categories indicated before, you can see at a glance that now the activation in the brain, and I should say these are flat maps of the brain. This is the temporal lobe here, parietal and prefrontal cortex. You can see the broadest activation that we've seen so far. And what was particularly interesting is if you're now looking into these regions that are selectively activated by social interactions, there are some regions that are actually only activated when the monkeys are watching social video. So I'm showing here 2 areas, one in medial prefrontal cortex, one in dorsal medial prefrontal cortex, areas 32 and 9M. You see that there's only activation here during social video. There's no activation or even suppression of these areas when you're showing any other kind of video. Now this is reminiscent to the default mode network in the human brain that is more active when the subject is lying in the scanner, not having to perform a task, than when the subject is seeing things on a screen or is performing a task. And it's often been assumed that this is happening because now subjects are mind-wandering, and when people are mind-wandering, oftentimes they think about social things. And so it's a possibility that this is a precursor of high-level social cognition areas in humans. And these areas are, again, like in medial prefrontal cortex, dorsal medial prefrontal cortex, ventrolateral prefrontal cortex, the parietal lobule, and the temporal pole. These are all areas that also in humans are being activated selectively when humans have to make these mental state attributions to others. Okay, so what have we learned from interaction analysis? Interaction analysis is ingrained in the whole perceptual system. So I told you initially we thought maybe some part of the face processing system is engaged, maybe the high-level areas are engaged. More likely I thought that the system outside of the face processing system would be engaged, but it turns out it happens from the very beginning of the face processing system. I told you that face and body areas have different specializations. We're speculating that maybe the body areas are feeding into the mirror system because the function is similar to the mirror system. that the face areas are feeding into the social brain because they have some functional similarity, but we don't know the substrate of these arrows yet, or if this is correct. But what it might mean is that our way of thinking about face and body areas so far might have been too limited, in that these areas might be doing more than just analyze facial shape or body shape, but that they are stepping stones to more high-level social cognitive tasks. We found a new function for the mirror neuron system. We found that large parts of the brain are engaged in social analysis. These are areas that correspond again to human areas that are involved in the most high-level form of social cognition, theory of mind. And what this means is that we can now, we talked about this over lunch, is these videos that we used so far, they were natural videos, but we can now use animation, so we can systematically decompose this animation to figure out what are the critical features that are necessary to get these social interaction areas activated. And so we can now process at least some forms of high-level social cognition through a rational, vision-based strategy. And finally, what does it mean that you understand social interactions? When you understand a social interaction, it means that you know who is doing what to whom. You know the subject of the action, you know what the action is, and you know who's the patient of the interaction. So you have to have some kind of language-like representation, maybe in the form of language of thought or other kinds of structured representations. But it means more than just an association, that you know that A and B are present and that there's an action happening. You know who's the source and you know who's the sink of the interaction. So the bigger point that I want to make here, just as the face processing... Okay. Sorry. I did not connect my power supply. Yeah, so it might be the battery of my computer. Do you have an adapter for US? But I might have one myself. I'm terribly sorry. I have one, yeah. But where's an outlet? Okay. Okay, so the bigger point I wanted to make, actually, how are we doing in time? Oh, thank you. Actually, I have a connection now. Thanks. How are we doing in time? Okay, so the bigger point I wanted to make is that we, because of the specializations that exist in the primate brain for social perception, for the perception of faces and bodies, we can use the system as a model system to elucidate general principles of object recognition or of feed-forward processing and other general processes in the brain. And because similar specializations appear to exist for other forms of social information processing that we can tap into by looking at the responses to social interactions, we can also now use it as a model system to get at other bigger questions, like the representation of structural representations of information that is compositional and can occur in any kind of possible form. Okay, second brief point I wanted to make was about personal and social knowledge. So I showed you that when you see the picture of Charles Darwin, you cannot not see Charles Darwin, you have to recognize them. There have to be links between perception and memory in a way that certain visual stimuli are activating your memory. And so we're investigating this, looking at the difference between familiar and non-familiar face recognition. So what's the difference between you seeing a picture of a personally familiar individual or the picture of a stranger? There are different hypotheses I'm not going to go into, except for one, is that we're thinking that maybe there's an extension to the core face processing system that I showed to you, that maybe there are extra face areas that are dedicated to the processing of familiar individuals. If they are modular, that is, if there's a clustering of cells, we would again be able to detect this with fMRI. And so Sophia Landi was an amazing grad student in the lab she took on this project. She mapped out the face areas that I showed to you before. And now, when she contrasted personally familiar items, personally familiar faces with personally familiar non-face objects, she found two areas that we had not recognized before. One is located in the temporal pole. The other one is located... This is personally familiar faces versus personally familiar objects. So she finds these two extra areas, one in the temporal pole and one in the perirhinal cortex, that are activated by this contrast. Now, this could be for a number of reasons. It could just be that these are more powerful stimuli and that therefore we see areas in the brain that we have not seen before. Is again not to say that these areas are not activated by non-familiar faces. They are, but not as strongly as by familiar faces. So what Sophia then relied on in the second experiment, and it's a critical one, is a deblurring technique where you're starting with a stimulus that's so highly blurred it's even difficult to know that there's a face. So I want to demonstrate this to you by giving you two examples. So I have a highly blurred stimulus here, which is now deblurring slowly over time, and when you recognize what is shown here, shout it out loud. So by now probably all of you will recognize that it's a face, and then someone will recognize that it's Obama, right? So there's this moment, eureka, aha, I know who it is, and it's someone who I know. So here's another example. The other side. Martin Luther King or Mao. Okay, so you're very eager to see someone in the picture. I'm quite sure you don't know who it is. Am I even deblurring it? Yeah. I haven't even started deblurring it yet. Okay, good. Sorry. Okay, so most likely none of you knows him. He's the half-brother of Obama. He became notorious during one of the elections, but so you don't know him. So the idea is that there's a fundamental difference between the processing of personally familiar individuals, or sometimes you might see someone in a picture who's not there, like Martin Luther King. I think by now you know it's not Martin Luther King, but there was a possibility for him before. So the rough idea is this, that if you have these stimuli that are very similar to each other and don't have semantic meaning in the beginning, and you slowly deblur them, if it is a generic face processing area, it will just increase its activity level because there's more and more information coming in.
What do you mean by non-familiar face recognition?
So, okay, so you can tell, but
what does it mean?
These are pictures of individuals that are not personally familiar to the...
What is the task?
Yes, so there's no task. There's just...
What does it mean to recognize non-familiar face? That is their face?
Right, so, okay, so the terminology is a bit... You could use like a different... We can talk about perception. So there's not a task where I have to say, this is this person, this is that person. There's also one use of the word of recognition, that it is always about particular individuals. This is not how I mean it here. Let's say like face processing. So we can process information from faces that are not personally familiar, and we can process information from faces that are personally familiar. That's all I mean here. If it's a generic face processing area, maybe there are differences, but there should be a more or less linear increase in activity as more and more information is being presented. While if it's a familiar face processing area, there might be nonlinearity at the moment of recognition, so akin to the eureka moment.
So, for example, you could say for the non-familiar face, tell me if it's a man or a woman, how old is he, or some intermediate recognition task that for some time the blurring is sufficient that you say that's a man, it's an older man, or a white man.
Yeah, so you could do that. We didn't do it, but you could do it. Yeah. Okay. So what did Sophia find? So in red we are indicating responses. These are all the classical face areas. These are the new face areas. Red are the familiar faces, blue are the non-familiar faces, gray are the objects. You can see an increase in response as we're adding more and more information across the entire system. But only in the two new areas that you found, there's a very clear nonlinearity. And actually in area TP, there's only a response in this deblurring to the familiar faces, not to the non-familiar faces, and not to the objects. And in PR, there's a big difference in response here. And we can quantify this. This nonlinearity is significant for these two areas, not for the other ones. So what Sophia found are that there are two new areas that are selectively processing familiar individuals. We don't know yet. I talked a little bit about electrophysiological recordings we started to do in these areas. This is very early days. Like one idea that we have is that maybe the kind of representation of faces here is fundamentally different from the core face processing system, where I showed you we are thinking about the representation of faces in terms of face space. So you want to have a activity vector in your population that is pointing to different parts in this physical face space. While here for the familiar individuals, it might be more like an attractor space. That it really will depend on, being this person or is that person, and if you're morphing one face from the other, its activity is going to remain here longer until it flips over to another face. So these are things that are still need to be seen, but I just wanted to emphasize this to indicate that there are fundamental general questions about the interaction of perception and memory and representations of visual information that we can get at exploiting the fact that the system exists. Okay, finally, emotional responses, and communicative signaling. So I showed to you that when you're seeing another face, ideally a face that's moving because of the facial musculature that is making facial expressions, oftentimes you're mimicking these facial expressions with a corresponding expression. So if you see a smile, you smile back, you might feel like a little bit happy. And so our thought was that in a situation like this one here, where this is mimicking effect, or in the situation of a mother and a child, where there is this sort of second person interaction, like between an I and a you, that we could get not just at the recognition of expressions, but the illicit, generation of emotional response, and then finally the motor response of the facial expression. So we started this thinking about a feed-forward processing system, because we knew that the top of the face perception hierarchy, AM, is projecting to the basolateral amygdala, that the basolateral amygdala is projecting to two areas in the cingulate cortex, M3 and M4, which in turn are projecting to the facial nucleus, which controls the facial movements. So anatomy is supporting this feed-forward view, this kind of interaction taking place. You see something, you're responding to it with a corresponding smile, might indicate a kind of feed-forward processing. And human neurology is also indicating that these medial circuits in the anterior cingulate might be sufficient for the generation of an emotional expression, while the voluntary control is more lateral. Stephen Shepard was the postdoc who took on this project. It was very risky from the beginning, because we, of course, number one is we didn't know we would get anything. Of course, I'm talking about this, we did get something. But number two is, if there's one thing you don't want in the scanner, it's movement. And what you really don't want is movement right next to the brain where you want to get activity. So very concerned that face movements would actually mess up everything. They didn't. So what he was using were displays that he took with the video, where he looked at an actual social interaction happening between a subject and another monkey, either taken from the front, so this is a second person interaction, a second person perspective, or from the side in this third person interaction, where it seems to the subject in the scanner that here there's someone trying to engage with you through the generation of a facial expression, and here it seems that he might be trying to engage someone else who you don't see. So it's the second versus third person interaction. So when Stephen looked at the activation, this is across 10 different individuals, in this condition versus that condition, what he found is a very selective activation, very anterior in the anterior cingulate cortex. There's no difference in the face processing network, which would be here. It's only here in this very anterior cingulate region. So we think that this region is responsible for sensing that the social contact is now like an IU contact, where a direct interaction could be happening with another individual, different from this third person interaction that Julia found. And indeed, this area that
How do you control for the orientation?
so there is a difference of low-level visual difference, but because in the face processing system we don't have an area for like front view and an area for a side view, but the cells for these two things are intermingled with each other, we don't see a difference in activation here, or like in earlier parts of the visual system. So this was the map I showed you for Julia's interactions, third person interaction. This activation for second person interaction is at a slightly different location. But now what's happening, and this is really only happening during the first day or first day and a half when we're doing the fMRI experiment, is that while we're showing the second person videos, the subject monkey on 20% of the trials is responding to the video with a lip smack movement. This is a very characteristic movement the monkey are making. It's an affiliative movement that's indicating I'm friendly, be friendly to me, we're getting along just fine. And for about a day, just showing this video clip was sufficient to elicit this in 20% of the cases. Not afterwards anymore. The subjects then found out it has no consequence what I'm doing as to what I'm seeing on the video screen. But we could fool them for a day and a half, and this was enough for us to get this map of activation for which regions in the brain are activated when the monkey is generating this lip smack movement. And we have similar maps for when he's drinking, so two different kinds of facial movements. What we found is multiple areas, S1, M1, lateral premotor cortex, medial premotor cortex, areas M3 and M4, the two areas that we had expected. But really what we did not expect is that the strongest activation really is in these lateral movement areas, some of which are thought to be corresponding to Broca's areas in humans. And so it's very different from this view. And we also get activation in 3 regions of the insula doing this generation of this very, very simple stereotypical movement. So there are multiple areas engaged in this task. Again, these areas are different from this area that is selectively responding to the second person context. So finally, Stephen compared lots of things in these areas. So these different colors and positions are indicating functional specialization of these areas to the perception, to second-person context, and to the movement. And the strength of the connections between these different regions of interest are indicating the strength of the functional interactions. He also did independent component analysis. The brief message is we have a network for face perception, we have a network for the generation of facial movement, and there's relatively little functional interaction between them. All that we found is actually subcortical and not between the cortical areas themselves. And then we have this third network, again with weak interactions only to one of the motor areas that is selected for this second-person context. So the view that we have now of the brain is that there is a face perception network that I talked about initially. There's a network for generating this facial motor behavior, and then like a social cognition area that's linking these two. But the predominant mode of activity in these networks is mostly characterized by internal connectivity, functional connectivity, but not by connectivity between these areas. So we now have arrays implanted in these in 6 areas, 4 motor areas and 5 motor areas in this medial prefrontal cortex. And Ifat, who will be back in a few weeks, is actually working with me on the physiology of the control of facial movements. Okay, so my summary, just very briefly, is Buber was right in some ways. I just mean this as a very, very loose analogy, and please don't take this in any deep sense of the word. But his reasoning that there's a fundamental distinction between an I-you interaction with the world and an I-it interaction with the world might be mapping onto different parts of brain architecture. It makes a lot of sense to me that there is this distinction that because we are social individuals, we have to get this distinction right. There's a network for voluntary movements like drinking and emotional facial movements that are largely overlapping. We thought that they would be like distinct, one medial, one lateral. We find both active. There's some difference activation, but mostly they're very similar, including in area that's similar to Broca's areas in humans. There was one person that suggested before that the lip smack might be an evolutionary precursor of speech, which I never took seriously. The argument was the rhythmicity of the lip smack is similar to our speech pattern, and I don't think that's the strongest argument. But now actually it turns out, yeah, if you have lip smack movement, a homologue of Broca's area is active in the macaque monkey, and so this might give us some clue about the evolution of speech and maybe some inroads in understanding these kind of movement patterns. And because of this reflex between the perception and the generation of facial movement, we also think we might have a chance to understand the neurobiology of feelings. So this is the pattern of the different networks in the macaque social brain. The biggest point right now, I have only shown fMRI to you. I hope that when I'm back here, hopefully in the not too far future, I can show some actual physiology. I'm an electrophysiologist. It pains me to only show fMRI activation, but this was the first step, a necessary step to parse out the social brain of the macaque monkey. The bigger point is that I think we will have a chance to understand high-level social cognitive functions and mechanistic levels, just as we did for the face areas, because we have similarly specialized subcircuits in the macaque brain that we can map just as we could map the face areas with fMRI. So I would like to thank the wonderful people in my lab, my funding sources, Rockefeller University, my home, and you for your attention. Thank you.
So we have time for a few questions.
Yeah, I was wondering, when you're talking about social interaction,
Yeah.
one thing that you may want to think about is sort of all the techniques that Dr. Malach and Uwe Hasson used,
Yeah.
where they just reverse the video and
Yeah.
show it backwards.
Yeah.
And I think that in that case, the social context is much less clear.
Yeah.
And it's nice, or you could scramble the
Yeah.
the video images and check that, and then parse out low-level issues that Chaim was worried about.
Yeah.
From higher level.
Yeah. So I actually agree. So for the electrophysiology, we have thought about the reversal. There's several reasons why you might get these activations, and so some could be that you're just trying to infer what's happening physically, right? And then another way would be to infer like the reasons for why what's happening is happening. Another way would be to think about like social scripts, right? Like is what's happening within the realm of possibilities, like if a tiny monkey is now threatening like a bigger monkey, it should never happen, and so on and so forth. Is there a prediction of what's going to happen? And so for this, as in speech, you could just reverse the video image or scramble it. We've done it for faces before, and it's a very powerful control there. Here we didn't do it yet because we don't know yet. It might be sufficient just to see a snapshot of an interaction to infer what's going on. So we don't know yet even whether movement is at all necessary. So like an iconic image of a big monkey threatening to bite a smaller monkey might be enough to get these areas activated. But it might also be that the motion is important and on different time scales possibly. And so like scrambling on these different time scales would be, I think, a very important technique.
Wondering about what you think could be extrapolated for, you did all your research on faces,
Yeah.
familiar and unfamiliar.
Yeah.
About familiar and unfamiliar objects.
Yeah.
For example, I work a lot with radiologists.
Mm-hmm.
I see how they have this gestalt
Yeah.
type of recognition. Here's a tumor, and it's a bit like you're blurring examples.
Right,
which
right.
if I come and see and not trained to that.
Yeah.
Do you think there's some encoding of some special object features which is not specific
Yeah.
to the face?
So yes, there is. So there are more results that Sophia had that I did not talk about. The interesting thing was that there's a fundamental distinction in how familiar faces and familiar objects are processed. So in the face processing system, personally familiar faces are enhancing activity. In the object selective regions, personally familiar objects are reducing activity in the object regions. We thought a lot about confounds like emotional factors, arousal to the stimuli, but we found no evidence that this is an explanation. And so it could be in line with work in olfactory and other systems that as you are more and more familiar with items, that your presentation becomes more efficient, and so therefore quantitative activity might be lower. But this is a speculation at this point. What we have not seen, and in other work we tried to look at this, is sort of a recruitment of face areas for the processing of familiar non-face items. So there's been a thought that these face areas are really only engaged not because they're faces, but because the stimuli you saw in early life are being presented, or the stimuli you're most interested in are presenting. Different theories of this sort.
So they're only temporarily engaged for that specific task and time, but there's no...
So depending on your ontogeny, you might change this. So Marsh Livingston's now looking at the ontogeny of these face areas and like when do they emerge and when they are there. What she has found is that when she trains monkeys on discrimination of numerosity, when they have to estimate which is larger than the other, and then replace numerosity with alphanumerical symbols, she finds activation just ventral to the face areas. So these now very, very familiar, specialized, meaningful objects that are not faces. So I think there's something to the larger area being, specialized maybe at different periods of time, doing ontogeny on different things. But when they're trained on these things like a human expert would be, they develop, but only when it happens very early in life. So when they're older than 2 years, which might be like puberty and puberty age in humans, then there will be the specialized areas. Afterwards, she doesn't see them anymore. So I think the perceptual experience could also be very different in humans, or the proficiency of processing these symbols could be very different in humans depending on when they learn. So the professions that you talked about likely after puberty, but yeah.
So one question about the fMRI studies, the last part. So how would you check causality in this? I mean, there is no task here.
Yeah.
So
yeah.
is there a way to manipulate and
right.
there is something that's consequential for...
Yes. So this is, for me, is a fundamental problem. So we have the discussion in the lab a lot. So my motivation for all of these studies was automaticity. And it was in part a pragmatic argument that I thought we had a chance to use faces to learn something about the organization of other brain circuits that are involved in high-level tasks, and that this would work automatically.